presented to us. So what we are doing to overcome all the challenges that in general we are facing in the research community is, first, we are ensuring that, especially for the archetypal side that we are leading in combination collaboration with the foundation, all those cells are hosted at the Center for Regenerative Medicine Bank. That's a report that is sustained by 10 brilliant, amazing, and well-funded PIs, and we've got institutional commitment. So those cells are safe and secure beyond the life of a working lab. The second thing that we are also doing is to focus a lot on training those who are going to take care of your children and the children of your children. That's why Sandeep is actually going to be presenting this talk. I'm focusing to make sure that we can continue doing training to these scientists in a patient-partner manner, which is something that we need to do. And the third thing that we're doing is not giving up. If there's something that I learned from you, is that no matter what challenges are, we're going to continue pushing and moving forward together. And I want to bring back what Jordan said about the synergies and the umbrella things. I do think that it's extremely important to characterize very well mechanistically things for clinical trial. We do basic science to understand the synergies. How can we study this in a combined manner at the basic science level? And we're hoping that our results can help the more applied and mechanistic and clinical studies to be pushed to clinical studies. And with that, I'm going to just get Sandeep give the presentation.
Thank you for that introduction. Hello, everyone. Thank you for. Oh, awesome. Just. So today I'm going to be talking about some work that our lab does with KAT6. The talk title, What KAT6 Blood Cells Reveal About Development and Disease Course. To start, I just want to go over the main takeaways I want you guys to understand in the talk. The first being that I want to convey that you're learning about from the epigenetic information in KAT6 blood cells that we derive from the triangulations right here. And what they can tell us about what happens during development in the womb when these cells are with KAT6 variants as they're generating the different organs that arise and developing what's going on there, and what these blood cells directly may be able to inform us about the pathology of disease. And the second thing is that understanding the biology of KAT6 and moving science forward is best done when working together. Where you're started on this topic. I want to thank you guys again so much for the donations you kind of gave in the perfect story. Okay, as I moved here, it all starts with the PBMCs. These are the peripheral blood mononuclear cells that exist in blood vials and stimulated. With these cells, we can reprogram into cells called induced pluripotent stem cells. I'll go into a bit more detail in some more slides, but just as an overview, I want to introduce the concept that we're studying development through these induced pluripotent stem cells. And these have the power to turn into cell types of various different organs, allowing us a huge potential to model different same disease in different organ systems. The second thing we're doing with these PBMCs is looking ahead towards the future. What can these blood cells directly tell us about KAT6, its phenotype, its current existing phenotype, or its potential disease course? And this lies in the epigenetic information in these cells. So this is using the histone post-transition modifications that occur based on the presence of KAT6 and other histone modifiers. And so understanding this fingerprint that arises from the epigenetic landscape potentially inform us about what's going on with the variant. So starting with a bit of a metaphor to go back to basics, what is the means of KAT6 sufficiency? I want to bring the metaphor of engineers working on building a structure like a house. And to relate to this metaphor, KAT6, these two engineers make two copies of KAT6A or two copies of KAT6A. And together they work to build together the structure of a cell, or going one step ahead, the organ and the cell. And how these engineers might help build this house is in this metaphor. they will highlight certain parts of the blueprint. So this blueprint has more information than what is needed to build a house, for example, in this case, 3 windows. But they'll highlight only 1 window and 1 door to build this house here. In the same way, with KAT6 and KAT6 they modify the histone marks on histones that are wound up, that genes wound around. And these highlight which genes are going to turn on, which genes are going to turn off, modify gene expression. So they work on top of the blueprint. They don't modify the existing genes or blueprint, add on top of it what's going to be expressed and what's not going to be expressed. And so in the KAT6 or KAT6A, KAT6B epigenetic variants, there's a loss of function of one of these engineers or one of these proteins, one copy of the protein, sorry. And in this analogy, that removes one highlight, and in this case, there's no highlighting of this door blueprint, and so the door is not built. Like, it's a bit more complicated when we look at a cell. There's a lot more than just 2 engineers, hundreds of histone modifiers working together in different ways, and how the loss of KAT6 variants impacts the histone landscape is not entirely known, and how that impacts gene expression is also not fully understood in different cell types. But we do know that it leads to different cellular phenotypes, as explained by previous talks, and this ultimately leads to differences in the full person. And so in this work, we're trying to understand the first step. What is the difference in these histone modifications that's being caused by the haploinsufficiency, the loss of function of one of these KAT6 alleles? And in doing so, we can uncover what that fingerprint is, this unique identity of KAT6 loss of function cells, their epigenetic landscape. So in order to do this, we start with, once again, I'll come back to the PBMCs. And we're trying to measure what this unique fingerprint histone landscape is using antibodies against different histone marks. So in this example, we just have this green colored antibody that binds to one type of histone mark, and a purple antibody that binds to a different histone mark. So it binds with all the histone marks throughout the cell as appropriate, and shine a certain color. And we can measure that color based off of its fluorescence. So the more, in this particular example, there's a little bit more green antibody, so it's a little bit brighter on the x-axis, which is fluorescence intensity. And then in the case of purple, there's a little bit less, so it appears, this whole cell appears a little bit different. When you take into consideration the entire PBMC population, you can get an entire spectrum, multiple cells at different fluorescence intensities. And it can change based off of course, the influence of KAT6 haploinsufficiency in these cells, but also the environment of the cell, what kind of cell type it is, what particular peripheral blood mononuclear cell it is. What is the cell actively doing? All these factors contribute to this unique fingerprint that appears when we take the entire PBMC pool into consideration. And what this fingerprint might be able to tell us, at least in the future, is potentially what the existing phenotype is in the patient. For example, if a patient has more neurodegenerative symptoms, then perhaps that fingerprint will reflect that. And even beyond that, it might potentially tell us more prognostically what we might expect symptomatically. But as far as we can tell right now, we're focusing on the basic science. We want to understand, first of all, what is the fingerprint? Is there a unique one in KAT6 patients? And beyond that, is there a unique one in KAT6 patients with certain symptoms? However, these samples are quite rare. And so I'm going to show you some data just on proof of concept PBMC and fingerprint data just on control cells. So starting with this is a unit that shows each dot is an individual cell. Based on how close the dots are, indicates how closely related they are. And related they are, I referring to taking all features that we measure into consideration. This could be cell lineage, what kind of cell they are, what the cell is doing, and of course the histone marks, that unique epigenetic fingerprint. Taking all those factors into consideration, it groups these cells together and forms clusters. And that's what the exome clustering is. It's what the computer tells us these cells are relatively similar, so we're going to put them together and move. And so this allows us for a very unbiased, unsupervised method of categorizing cells together. We can apply our own prior knowledge to understanding what these clusters actually are by looking at well-known lineage marks of different cell types. So if we look at, for example, CD45, which is a pan-glucoside mark, we can label that one color, and B cells sequence with another color. What we can see quite strikingly here is that different cell types arrange themselves quite differently. So, for example, in this population right here, this green group is very far away from all the other cells. It indicates that across all the factors that we probe, they seem to be quite different from the other ones, indicating that they might have their own phenotype. Moving on to what the cells are actually doing, we're looking at this is their cell cycle phase, G0 and G2. This is just how much they're dividing or actively engaging in the cell cycle. And you see that there's differences here as well. And we're able to regularly capture that within this PBMC population. This gives us a quick spread of seeing different types of cells that have different behaviors, giving us a good idea of what the whole picture is. Finally, the main point of doing this method is to understand that the histone modifications are globally across these different cells. And so here on the x-axis are all these 18 different cell clusters, so these are just groups of similar cells. And you can see that the abundance of one particular marker, in this example, H3 K27 acetylation, differs across these groups of cells. And this is just one histone marker. When we put multiple together, you get an entire spectrum. This is showing, for example, some marks of how the cell is actively doing in terms of apoptosis or cell cycle, but all of these are histone markers here, these 6, and these are 3 different clusters. This is not the PBMC data, this is just example data, but the main idea here is we see a unique fingerprint appearing in each of these different clusters. And now we can start to extrapolate what does this mean for the cell in terms of its actual phenotype. How does the fingerprint relate, and what can that tell us beyond the cell itself about the disease course? Creating, like I said, a full picture, a fingerprint of KAT6. And so our eventual goal is to use multiple different PBMCs for multiple patients with different symptoms and trying to understand what the unique fingerprint is of KAT6 as a whole, and then what the fingerprint may be for individual KAT6 symptoms. The utility of that is if we can identify a fingerprint of KAT6 as a whole across all patients, what's a general feature that's shared among KAT6 patients, that could potentially be used as a biomarker when we use therapies and see testing if a drug works or not. We can apply that drug and measure these readouts and see does their fingerprint start to return back to a control fingerprint, or does it remain as this unique KAT6 fingerprint? And then beyond that, if we can get a symptom-specific fingerprint, then we can potentially use it as a prognostic method where we read the patient's PBMC fingerprint and predict will they have more severe gastrointestinal issues or more severe in the future features. So those are the 2 potential uses of this technology, but we're still very early on. And before we can understand where the cell is going, we have to first understand how we got there. So I'll return back to our developmental studies. And for that, it comes back once again to the PBMCs. But this time, we take the PBMCs, and this is our actual exact flow that we'll go through tomorrow, probably, is we're going to cryopreserve peripheral nuclear cells, and then at a later time when we're ready, we can thaw them and use them for reprogramming. And so we'll go into a reprogramming pipeline one at a time, where we deliver plasmids that contain genes that will push these cells into what we call an induced pluripotent stem cell, which you've already heard about a little bit, but just to reiterate, induced pluripotent stem cells resemble very early stem cells in development. When the baby's developing a fetus, these early on stem cells have the potential to turn into any cell in the entire body. And so we recapitulate there, we induce it, that's the induced pluripotent stem cell, to recreate that cell and have the ability to model any cell in the body, any organ, by using, but before that, another key feature of induced pluripotent stem cells is that they have the capacity to indefinitely self-renew. And what that enables us to do is grow these cells up and then take a portion and freeze it down. And then with the remaining portion still alive, you continue to grow them theoretically for an indefinite period of time, creating a pretty large pool of these very powerful stem cells. And all the ones that we freeze down periodically over and over again, we can then send them out to other KAT6 researchers across the globe that may be interested in modeling different organs that we're already looking at. And that way we can expand the science a lot faster, giving access to these very rare induced pluripotent stem cells to multiple groups. Just as an example of what we've done so far, this is our Center for Regenerative Medicine, they have a motto for open science and sharing in these induced break bone stem cells. So right here is a figure showing that these historically over the lifespan of the Center for Regenerative Medicine, all of the induced break bone stem cells we've shared across the whole globe. These are not KAT6 regular samples, that's relatively recent. This is all the iPSCs we've created, and we had a lot of success in sharing ourselves. Looking at KAT6 in particular, we started back in 2022 collecting samples just at this conference, and we had success largely, I mean, completely actually, thanks to your guys' kind donations of PBMCs, and every year we collect more and more PBMCs for the programming. This has given us a large repository of different variants of the KAT6 and KAT6 gene. And so all of these are PBMCs we have been via frozen, and we're going through the process of programming. Already we've programmed several cell lines for both KAT6 and KAT6, and we have a lot more that we're excited to go forward with. But what are we doing with iPSCs? Well, we're trying to recapitulate development. To do that, we give these induced break bone stem cells very early young primordial cells the same factors that they would receive during development in order to mimic the developmental course. And in doing so, we can create these organ-specific cells, such as in this case, a neural progenitor cell. With this cell, we can then look at the same epigenetic fingerprint, looking at the histomites these cells of a particular cell type now, not the ADCs, but the neuron itself. So we can identify what that spectrum, epigenetic landscape is going to look like. What then we can ask what will that tell us about its existing state and potentially how the disease has progressed to the point to produce that cell back time. Essentially indicating how KAT6 influences the epigenetic landscape during development. Finally, with all these different cells we're able to create, we can also probe different organ system models. Using the cryopreserved cells, we can model them, instead of turning them into progenitor cells, we can give them factors to look at a different organ system. In this example, we're looking at gut cells. And through this power, we're able to look at essentially any feature of the body that we think is most scientifically important to investigate. So I'm going to recap the goals of the Serrano one, as far as the KAT6 area is. And that's one, looking at development past, understanding how these cells came from by recapitulating development in a dish using induced break bone stem cells. And the second area is looking into the future and the present by directly probing the PBMCs to understand what their unique epigenetic fingerprint is, what that can tell us looking ahead, what symptoms might appear, or creating KAT6 as a whole as a biomarker. And for that, I want to acknowledge the Serrano lab and many different institutes of the Crown that have helped push our research forward and enabled us to share these ideas around the globe, and we program the PBMCs that you have kindly donated and actively doing so. And so that includes a bioinformatic program, our collaborators, including the current founder, Daryl Cotton, we're programming for led by Mary Ann, and some of the other institutes we work with. I also want to give acknowledgement, just to let you guys know, some collaborators that help advance the science and really push it forward into novel and exciting ways. With the Chen lab, we take the cells, these iPSCs, we differentiate them into vascular cells. With those cells, we can look at very high throughput and very informative features using their bioengineering devices. With Dr. Rosenthal, we're looking at placental cells, applying this EpiFlow into other avenues of diseases. And finally, introducing Prorizula, where we now use an in-vitro model of the zebrafish to understand how epigenetic modifiers, the loss of function of epigenetic modifiers impacts the phenotype, and now follow the finally, I want to acknowledge the Gestapo lab recently got funding for a foundation where we're looking at the epigenetic landscape that I mentioned previously, as well as the gene enrichment networks in and testing for human cells in specific KAT6A and KAT6B disorders. Finally, I would like to acknowledge the most important group, which is you, and all the kind donations that you guys have given, you're giving next door through the PBMCs, and through that, is really the big picture where we're able to move science forward well beyond this particular project. Through the IPSCs, we can share them throughout the world to future scientists and really build a community of scientists for KAT6 that really push science forward. Thank you so much. Okay, thank you. Any questions for this presentation before we take a 10-minute break?
If we have deleted in the past, what we have to be to again is equal to again, or do you have to be?
Yeah, so you absolutely don't need to, but we would definitely appreciate that because as far as the induced peripheral stem cells goes, once we're able to successfully recruit them into iPSCs, as I mentioned, they can repopulate indefinitely, so that gives us a solid pool. However, not all the time, every time we do PBMC successfully in the program, it's fairly consistent and reliable process, but not 100%. So in cases where it fails, we might need more PBMCs to draw back upon to try again. And that's at least looking at development. Looking into the future and how analyzing the KAT6 fingerprint PBMCs, we directly probe the PBMCs themselves. So in that case, we don't have this indefinite self-renewing population. It's just the ones that we have donated. So for that reason, any PBMCs blood that's donated will help.
Thank you so much. One question that I have is the wild type IPS cell itself has such a deep variance between samples, and particularly from the landscape of how epigenetic vectors are changing. And based on the previous presentation, it's very clear that there are very clear, I guess not clear, demarcation of early transplantation rate, transplantation, and all this different categorization. I presume that it will significantly impact how epigenetic vectors is globally there. And I'm wondering about whether your strategies to make sure that you will be able to capture the biological significant differences in difference of types of patients as compared to the potential. Thank you.
That's a great question. So there's several different ways that we can try to understand whether this fingerprint, which you are looking from the epigenetic histone landscape, there's a lot of factors that play into it, and so having as little variability as possible is ideal. So there's other strategies we can do to overcome that. One, by having a large pool of KAT6 variants, we can pick out so separately. As you can figure, we have pathogenic variants at various areas on the KAT6 protein. In addition, we can use controls like patient short ones, where we take the same genetic background and just CRISPR edit the pathogenic variants. And that gives us a really powerful control where different cell lines based on having widely different genetic backgrounds are going to have, presumably as well, a different histone landscape that sincerely. So if we correct it, we can reduce it as much as we possibly can by having the entire rest of the genome work, essentially. As to your point of the variability in IPC differentiation, that's absolutely true and has been a problem in the field generally where different differentiations can look quite different. So to overcome that, we want replicates, triplets or even higher, to be independent differentiation. So we don't just take one differentiation, go into three, and say what are the differences. There we do three different differentiations. So that can try to control for the variability that we see across IPGCs. Okay. Thank you very much. Okay, we're going to for 10 minutes.